And so our conference ends with that beautiful word, hope, because now we're going to find out about the future metastatic treatments. What can we expect in the next 5 years? So that is hopeful for all of us. And I'm very honored to present the 4 doctors who are going to speak to us about that. First, Dr. Hyman Muss is currently professor of medicine at the University of North Carolina, Chapel Hill, and director of geriatric oncology at the Lineberger Comprehensive Cancer Center. His major research interests are breast cancer, with emphasis on the treatment of older women, issues related to treatment of all older cancer patients, and providing education in geriatrics to fellows in training. Dr. Muss is currently co-chair of the Cancer in the Elderly Committee for the Alliance, a National Cancer Institute-sponsored cooperative group which endeavors to increase awareness and clinical trials opportunities for older patients. Our next physician is Dr. Larry Marks. Dr. Marks is the Dr. Sidney K. Simon Distinguished Professor of Oncology, Research Professor, and Chair of the Department of Radiation Oncology. Dr. Marks received his medical degree from the University of Rochester in New York and completed an internship at Sinai Hospital in Baltimore, Maryland, and a residency at Massachusetts General in Boston. His interests include clinical trials in breast and lung cancer and understanding the determinants of radiation associated to normal tissue injury. Dr. Marks' work assesses the use of advanced technologies to improve the therapeutic ratio for patients with cancers of the breast and lung. He is also interested in errors in medicine and applies industrial engineering concepts to improve clinical operations and safety. Next, we have Dr. Elizabeth Claire Dees. Dr. Dees has been a medical oncologist treating breast cancer patients and conducting clinical trials of new treatments for metastatic breast cancer for over 15 years. In addition to her MD degree, she has a master's degree in clinical investigation from Johns Hopkins with a focus in novel trial design. She leads the Phase 1 trials group at UNC LCCC. She is also the co-leader of the clinical research program at the Cancer Center and oversees the core research that supports clinical trials operations. She has been the principal investigator for over 50 clinical trials. She and her group have a track record of conducting early translational clinical trials, including first-in-human trials of new anti-cancer therapies. And lastly, we have Dr. Nancy DeMoore. She is a professor of surgery at UNC and a member of the UNC Breast Center. With Cam Patterson, MD, MBA, she has co-founded NC Therapeutics Incorporated, a startup company that is developing a monoclonal antibody therapy for cancer. She has studied angiogenesis since her undergraduate days at Wheaton College in Massachusetts. After earning her medical degree from the University of Health Sciences, Chicago Medical School, and completing her internship and residency at Boston University, Dr. DeMoore completed a surgical research fellowship in the lab of renowned physician scientist Judah Folkman, MD, of Children's Hospital in Boston. So let's give them all a welcome.
Well, you're an incredible group. First, this is just a terrific conference, and I think all the physicians and staff involved have been overwhelmed to see all of you come, support the conference, be advocates for your cause and our cause in breast cancer research. It's really terrific. And there may be some men here with metastatic breast cancer. I don't know about that, but there's a lot of men here, and I assume a lot are spouses and caregivers. And my wife, who was early this morning, who's very interested in patient and family caregiver care, remarked that all the energy and strength and support in this group. So congratulations to all of you for getting together, for coming here. I know Dr. Anders will be thanking everybody, but Shirley Mertz and Deb Tincher have done a great job. And Carrie Anders, one of my partners and really an expert at metastatic breast cancer, has put her heart and soul in helping organize this conference. So I hope you really enjoy the UNC experience here. And Carrie, thanks for your great work. It's really spectacular. We have a great panel here, and we've had some great introductions. And what's really special about this panel is you would really enjoy being cared for by any of the doctors here, even on this panel. Even though they're all experts and have incredible CVs and have done all types of wonderful research, they're all outstanding caring physicians. And so I think it'll be very special to hear their perspectives. And these are the topics we're going to cover. Dr. Marks is going to talk about radiosurgery, proton beam radiation, and other new therapies in radiation oncology. Dr. Dees, who's really an expert on phase 1 trials and new drug development, is going to talk about some of the exciting new targeted therapies. Dr. Damore is going to talk about a topic of surgery in metastatic disease. And I know there was some of you were to talk a little earlier, but there are a lot of exciting things and opportunities for patients for surgery in the metastatic setting. And then last, I'm going to beg for money. And I'm going to ask you all to help us get Congress to give us more money for research in metastatic breast cancer. So I point out we're going to follow up on that wonderful earlier presentation by Rebecca. And this is really, we're going to answer questions about hope. We're going to tell you what's new and exciting and some of the opportunities we see coming. And we're only going to have 5-minute presentations. So we're going to have lots of time for questions and discussion. And we hope you'll really take advantage of us and challenge us with your questions. So I think we're going to start off first, follow the order here with Dr. Marks. You want to come up here? Yeah. We're short, so we're nervous about standing behind us.
Speak for yourself. It's a pleasure being here. I'm Larry Marks. It's inspirational to meet with some of you and hear your stories. It really is very humbling and inspiring at the same time. I'm a radiation oncologist. I think of myself as a surgeon. We have both local modalities. The hot new area in radiation oncology over the last several years is this thing called radiosurgery. It's the ability to deliver very high doses of radiation to small areas in the body. And we've had this ability for brain tumors for probably 15, 20 years. But over the last couple of years, we've now got this ability to do this in most every place in the body. So if you have, a solitary lesion in the liver that you might otherwise want to remove, a single metastasis, but you can't take it out because of its location, or you're not a good candidate for surgery, for example, you can deliver very high doses of radiation to a small area in the liver, or a small area in the lung, or really a small area almost anywhere in the body. Not everywhere, but most anywhere. And I like to think of cancer as a local and a systemic problem. Metastatic breast cancer, by definition, is a systemic problem, but there are deposits that we can see, and there are microscopic deposits that we can't see. So I think there's a synergy between the systemic therapies. As the chemotherapy gets better, the targeted agents get better, the biological hormonal therapies get better, and when our medical oncologists are able to sterilize microscopic cancer that we can't see, doing that concurrently and synergistically with radiosurgery for the spots we can see conceivably provides, I think, a fair amount of hope that there's a subset of patients who have limited metastatic disease that we potentially can cure, if not cure, give them a long disease-free interval. There's a track record for this. Our surgical colleagues have a long track record in colon cancer of resecting liver metastases, in sarcoma resecting lung metastases. A proof of principle that some patients with metastatic cancer have what we call as oligometastases, or a limited number of metastases, and treating those aggressively might cure some subset of patients. And then when you fold in the better systemic agents, there's a hope that some patients, including some who have microscopic metastatic disease that we can't see, that we can cure some of them. So that's the area that I think that most radiation oncologists are most hopeful of, and there are multiple studies going on looking at systematically trying to treat patients with metastatic cancer with that approach. Proton beam is just one example of fancier radiation equipment. I'm not a great fan of proton beam. Proton beam does provide a better dose distribution on paper. The challenge is there are a fair number of uncertainties with proton beam, and once you fold in those uncertainties, the dose distributions with protons, well, they're probably still a little bit better than traditional X-rays. They're probably not as much better as we think they are. I don't think that protons is imminently going to cure metastatic cancer. Protons is just a more exquisite and a fancier local hammer and nail, if you would, that some radiation oncologists have. These are incredibly expensive units, and there's only a handful of them around the country, maybe about 10 or so units around the country. But these are spreading, and you'll see these opening up in multiple cities over the next couple of years. There's one being built in Manhattan. There's two being built in Washington, D.C. There's now one operational in Philadelphia and Virginia, one being built in Georgia, I believe. There's one in Florida. And 10, 15 years ago, there were two in the country. And now there's probably 10 to 15 over the next couple of years. And in the interest of time, I think I'll stop there. Okay?
So I was asked to talk about future systemic treatments for metastatic breast cancer, so I made some notes for myself. And I think what's coming down the pike in terms of new treatments for metastatic breast cancer is part of a trend that we've seen over the last 5 to 10 years, where the focus in developing new drugs to treat metastatic breast cancer has really been on targeted therapy. And I think that's a word that we use a lot, but it probably is worth talking about what we mean when we say that as patients or doctors. And so I like to think of this as an opportunity for people who develop new drugs to leverage what the scientists have learned in the laboratories about what makes metastatic breast cancer cell grow and divide and progress and spread. And what have they figured out about the enzymes or proteins or pathways that cause that growth to happen? And then can we drug those targets? Can we develop a drug that would stop that pathway or enzyme from going haywire in that tumor and turning it from a normal cell into a cancer cell? So there's been really an explosion of work on targeted therapies. And you had a talk, or some of you had a talk earlier today on therapy for HER2 overexpressing disease, for example. And so you know that in the past few years alone, there have been 3 new targeted drugs to treat HER2-positive metastatic breast cancer approved for use. And another example is that we've learned that adding the targeted drug everolimus to anti-estrogen therapy is helpful in terms of extending progression-free survival in women with metastatic ER-positive disease. So we're already seeing some of these new targeted therapies come to market and become FDA approved and widely used. There are many, many others that are in clinical trials around the country, and Jean Noe and I are going to talk tomorrow morning a little bit about clinical trials and how to find them and what questions to ask and which ones you or a friend might be interested in or eligible for. But many of these are many of these targets are enzymes that are called kinases. So you'll hear a lot about kinase inhibitors. A lot of these are oral and really a very different kind of therapy than the traditional cytotoxic chemotherapy that was really the focus of drug development and treatment for this disease when we all trained. So these kinase inhibitors, I'll mention a few of them. One particular kinase is called PI3 kinase, and we learned in a project called the Cancer Genome Atlas that this gene called PIK3CA is actually the most commonly mutated thing in human breast cancers. So it turns out that there's a mutation that turns this kinase, PI3 kinase, on. It stays on. It's an activating mutation of this kinase that's not supposed to stay on all the time, and that may be critical in driving the growth of metastatic breast cancer cells. And so there are a number of these PI3 kinase inhibitors in development. Most of them are oral. Another example you might have heard about is something called cyclin-dependent kinase, or CDK4/6 inhibitors. Big news about a CDK4/6 inhibitor called palbociclib, and I don't have any conflicts to disclose, which is currently at the FDA, perhaps will be approved in combination with an anti-estrogen, improves outcomes for women with metastatic ER-positive breast cancer. So there's another kinase inhibitor that's really hopefully going to make great strides. And then just a third area I'll mention is that there's been a lot of focus in new drug development on drugs that modulate the immune system. So what is it that a cancer does to prevent your immune system from seeing it as wrong and killing it? What can we do to unmask that cancer so that the immune system and the chemotherapy works better? So a lot of these drugs, you might have heard of PD-1 or PD-L1 inhibitors. So one thing that's clear, though, is that kinases and pathways that drive a cancer are not just one problem. There's sort of a web of these kinases, and I meant to send Hai a slide to show of all we know of all these enzymes that drive cancer growth. So it's not going to be just blocking one. It's going to be figuring out, if you block this one, what else upregulates and is the escape hatch for that cancer cell, and how do we need to combine these to really assure effective treatment. And so this lets me make a pitch for the need for biopsies of tumors, both at the time of diagnosis and also at the time of a resistance to a therapy. So at UNC, we have a scientist called Gary Johnson. He's the director, the department head of pharmacology, and he's figured out a method in the lab of looking at several hundred of these enzyme kinases all at once. And he's been able to see if you hit a tumor in a petri dish or a mouse or a human being with something called a MEK inhibitor, what other enzymes upregulate? And this has led us to design a trial that has a MEK inhibitor plus a different inhibitor to hopefully hit both of those pathways and really shut it down. So I know you know this and feel this even more than I do, but thinking about incorporating biopsies and allowing research biopsies really, I think, is going to move the field forward. And the last thing I will say before Hai gives me the hook is say I got lots of ideas for future treatments. I thought it was worth saying a couple of words about sequencing and genomics and tumor genomics and genome-guided therapy. That it's out there, everybody's hearing about it and reading about it. So what does that actually mean? Well, that means that in addition to figuring out what are the pathways that are off kilter in triple-negative breast cancer in general, what can we learn about what went wrong in a specific cancer? So we now have the capacity to do sequencing of genomic alterations in a particular person's tumor. There are many university cancer centers around the country that are doing this. We have a program called UNC Seek, MD Anderson, Wash U, to name a few, and there are some for-profit companies that are providing this as well. So the idea is that you would be able to sequence someone's tumor, find out what genes are aberrant, and maybe some of those would be something that would be actionable, that we would have a targeted therapy that would hit. Now, the caveat is that this is very investigational, right? Nobody has run a trial that has said, if I sequence your tumor and instead of giving you Taxol, I give you brand new targeted therapy X, will you do better than somebody just like you who just gets the Taxol? We've not run that trial and proven that yet. So it's very intriguing for people like me who develop new drugs that this might be the way to figure out and really personalize therapy for metastatic breast cancer. But I just want to be sure that I'm clear that this would be the sort of thing to participate in a clinical trial. At this point, we don't know if this is going to be the right way to proceed. I'll stop.
I should add, are we collecting all the questions?
So I'm Nancy DeMore, and I'm a surgical oncologist. And part of my job is I train residents and surgical oncology fellows. And there are three main things that I need to teach them. Number one is how to operate. Number two is when to operate. And number three is when not to operate. Okay? And those are all very important parts. And when we talk about surgery in people who have metastatic disease, we're really talking about when to operate, what situations would it help, and when not to operate. When is doing surgery going to have no effect on survival? And so in particular, one of the big questions is if someone has metastatic disease, does doing surgery on the breast, removing the breast tumor, removing the lymph nodes, does that have any benefit? In 2001, when I was a surgical oncology fellow, I remember a discussion that we had at one of our tumor boards. And every surgeon in my fellowship, this is at Sloan Kettering, would really only operate on a patient who had metastatic disease if there were local issues of concern. For example, is a tumor bleeding? Is it large and painful? Is it infected? Or is it near any nerves, such as the brachial plexus, that can cause problems with the arm? So really, for symptoms would be when a surgeon would be called in and asked for help. Over the past 10 to 15 years, with the improvement with systemic therapy, where patients are living longer with metastatic disease, two new issues have arisen. Number one, if someone's living longer with metastatic disease, is that more time to lose local control of that tumor? Okay, if someone's going to live longer, what if the metastatic disease is doing great, responding to that chemo, but the primary tumor is growing? It makes sense to do surgery if that's the only place that's growing. So sometimes we are asked in a multidisciplinary setting, would that be a time to intervene with surgery? The other issue that has really been highlighted over the past decade is it possible that removing the primary tumor would actually improve survival of the patient? So this was a question that was unknown. And there have been many, many studies over the past decade that were retrospective studies that looked back on what surgeons did, comparing the patients who had their tumors removed to those who didn't. And of course, in retrospective studies, the surgeons are the ones selecting the patients, and as any good surgeon, I'm going to select the best patients for my surgery in terms of, are you a good surgical candidate? If someone has metastatic disease and kidney failure and heart disease, I'm probably not going to operate. But if someone's young and healthy and 25 and, my heart is pulled, I may operate. Not just me, I'm talking about surgeons. And so you can see that there's going to be an inherent bias in these retrospective studies that surgeons may select patients who may have a better prognosis regardless of the intervention. So there has been a lot of discussion at national meetings for many years looking at these studies that some of them are showing 6 to 12-month improvement in survival. And many of us know if we had a drug that improved survival by 6 months, that would be a multi-billion-dollar-a-year drug for a company, right? So the question is, can we get that effect with just doing surgery? So the big news is 2 negative trials. But negative trials can be good because we don't want to harm people with surgery as well. So 2 prospective randomized trials were presented in 2013 at San Antonio Breast Conference. One was from India and one was from Turkey. These were randomized studies, patients with metastatic breast cancer. One group had surgery, the other group didn't. They were all, in one study, they were all treated the same with chemotherapy, and they were stratified, so there were equal number of patients who had one MET versus multiple MET. So the only thing that differed between these 2 groups was having a surgical intervention. Both of these studies showed no difference in survival. And so I think the prospective randomized trials have really shown that removing the primary tumor is not something that's going to improve survival. Does that mean that there's no role for surgery in metastatic breast cancer? Not at all. We practice in a multidisciplinary setting, and sometimes surgery can be the right thing for local matters, because local matters really can be problems. People can have problems with pain. They can have psychological problems. They're doing great, but they keep feeling this lump, and it's bothering them. So surgeons can help in terms of local control, but the major data is that it's not a survival issue. And although negative trials are disappointing, as I said, we need to learn when to operate and when not to operate, because all of our therapies, including surgery, can have side effects. We can decrease quality of life if a patient has one of those side effects. And so we need to have clear data. So I would much rather be giving Dr. Dees' talk on targeted therapy, because as a surgeon, I don't have a whole lot to offer. That's why I'm in the lab working on targeted therapy. Thank you.
So I just wanted to come back to a topic that's been touched on by many presenters, and that's funding. I think a lot of people, when they think about funding in the United States and the National Institutes of Health, some people feel we're doing well and we're putting a lot of money into research of all types. But probably here, this group is well aware that if you look at what's happened over the last 5 to 10 years, things like the National Institutes of Health and the National Cancer Institute have actually had dramatic cuts in funding. And some of you may remember the sequester, which kind of has come and gone, where there was a fixed 5% loss of funding. And just as an example, in North Carolina, where we have a lot of federal funding for research, that meant a 5% cut in all our research. And it actually led to a loss of programs as well as a major loss of jobs in this state. You know, we don't have it back. No one's talking about that now. We have other political issues. A lot of us are nervous in cancer research about the lack of funding. Probably at this point, to apply for a new research grant, to study something in the lab or to do a very exciting clinical trial, the chances of getting your grant funded out of the National Cancer Institute is about 1 in 20. 1 in 20. So I'm an older guy, and in the 70s and 80s, golden years, your chances might have been 1 out of 2 and 1 out of 3. And plus, if you didn't get funded, there was enough help and people to help you in the government to help you rewrite your grant and go back in again, and you had a good choice. And so what this has led to is a lot of our young and brightest people leaving the field, you know, going out of cancer research and getting other jobs. And it's been hard even for established, very high-quality people in academia, in a lot of the federal programs, to really have sustained funding. So quite frankly, when a doctor goes up and talks before Congress, they say, oh, those guys are always whining. They've really got to hear it from you. You've really got to advocate and work with your congressmen, your local people, certainly people in Washington in this Congress, and really push for federal funding for cancer research. And it's more than just cancer research, it's the whole National Institutes of Health. So if you look at really bang for the buck in tax money, there's very little that competes with the National Institutes of Health. Studies on polio vaccine, HIV, cancer, that they have funded over the years. And so we really need to do it. And a lot of the things that we learn from other disciplines, other diseases, cardiology, rheumatology, we find now we can help our cancer patients with them. Many of our drugs, for instance, damage the heart. And we've learned from the cardiologists how to prevent a lot of that damage. So it's really supporting the whole biomedical research enterprise. And you can really play a role. And then lastly is the Food and Drug Administration. They take a lot of hits. They're slow. They actually do a great job. They're here to look at our food. They're here to look at the quality control of our drugs. They do an excellent job, and they're woefully understaffed. Congress loves jumping on them when there's a problem with a drug. But the truth is, they are fundamentally involved in our cancer drug development process. And they have some very fine people in the FDA who are outstanding scientists and are really there to facilitate getting things going. So putting in a word for the FDA is really worth it as well. They really are partners here to help us. And then lastly, pharma gets a lot of hits from public, from Congress, but the pharmaceutical industry has really been responsible over the last decade in developing some of the most promising drugs we have. For instance, all the anti-HER2 directed drugs have really come from pharma development. This is very good because they've picked up the ball. It's also a reflection of the lack of money in the National Cancer Institute, which used to develop drugs and which doesn't have the revenue to do it or to do the trials that can take hundreds, sometimes thousands of patients to do studies. So right now we're capped off. And where I was able to do studies with federal funding with many of my colleagues for 2,000 to 3,000 women looking at treatments for early breast cancer, looking for treatments for metastatic breast cancer for hundreds of patients, we're kind of capped off now. And so we've lost the ability to do a lot of these studies. So I urge you to work with congressmen, with local people in politics. This is a bipartisan view. It doesn't matter whether you're a Republican or a Democrat. Cancer is an equal opportunity employer. It gets adults, children, people of all racial and economic backgrounds. So my plea is to put a word in. We're always whining in the medical establishment, but we really all need all of you. And believe me, you'll have greater impact talking in front of a congressional group or a local group than a lot of us in saying we need more money. We need more money in metastatic breast cancer research and in all cancer research.
Thank you.
So, thank you.
Some few questions for
Thank you. Wow, there's a lot here. So, as I sort these, we'll start out with question one. Can UNC sequencing be done on previously removed frozen tumors? Dr. D, so you might want to explain what sequencing is and how we do it, and then the purpose.
Can you hear me? Yeah. So, yes. So I think the question is asking about archival specimens, so paraffin embedded. So it doesn't require fresh frozen biopsy. It could be done on your specimen from 2005 that's in a hospital in Wilmington, and the pathologist can go out and get it and do the assay on it. The UNC sequencing program is a trial right now, a clinical trial, sort of setting up the logistics of how to do massively parallel, well, what they call next generation sequencing on hundreds of targets. And so it's not got the sort of turnaround time that is clinically relevant to people like you and the doctors who treat them. So the faster way to get a smaller panel of genes sequenced is to use a clinical or CLIA-approved assay in your local cancer center laboratory. We have one called the Solid Tumor Panel. It takes about 2 weeks to come back. There's a company called Foundation One. That's the one I know of, but I saw a patient in consultation yesterday who had a different company's printout with her. So you might want to search that. There may be companies that are very close to you, and they would each have their own description of what needs to be sent.
Okay, so while I'm sorting these, a lot of great questions here. Larry Marks, can you talk more about the evidence behind oligometastatic disease? And really, people want you to define what you mean by it a little bit in more detail, and give some examples, some more examples of where you might treat.
Okay, so the proof of concept has been shown. Nancy, help me out here if I forget one, but sarcomas for lung metastases, there's a literature. Patients with sarcoma who have 1, 2 lung metastases, if you cut out those metastases in a healthy patient without other evidence of metastatic cancer, you can cure roughly 20% of those patients. Okay, colon cancer, same thing, lung and liver metastases, limited number, 1, 2 lesions, that there's a tail on that survival curve of about 20%. There have been now 2 studies that I know of prospectively using radiation therapy for oligometastases. One at the University of Rochester, they treated about 125 people, up to 5 sites of disease is what I remember. And they had roughly a 25% survival at disease-free survival at roughly the 4 or 5 year point. The local spot that was treated was controlled about 75% of the time, but a large number of the women, or a large number of the people, developed metastases elsewhere. But they did have a tail on that curve, which looks promising. And there's another study from the University of Chicago, essentially the same study, a dose escalation study, is a fewer number of patients. I think their limit was 3 sites of disease, maybe it was 4 sites, some low number. And again, same sort of thing. They had a tail on that survival curve. So the numbers that people have looked at are usually up to about 5. The lesions need to be smaller than roughly, I'll say 2 to 3 centimeters, depends on where they are. Some of the limit is a biological one, because if you have 10 spots that we know of, biologically we know that the likelihood of us curing you by treating all 10 of them gets lower the higher that number goes. Also, it's a logistical thing. It is harder to treat 10 spots. It takes longer, it takes more out of you, it's more complicated, et cetera. So there's a logistic limit. As our technologies get better, that logistic limit is getting easier to deal with. We can physically treat multiple sites. The question is it worth treating multiple sites? So I guess if I had to put a number, I'd say maximum 3 to 5, up to 3 centimeters each, something like that. Does that help?
So actually, someone came up with a follow-up question here. It says, how good is the evidence for treating oligometastasis with radiation, let's say, versus systemic therapy? And who's doing it, which I think is a more generic answer.
I think they're synergistic. I think the likelihood of having a success with this approach is in a patient who's responding to the systemic therapy, such that the other sites of disease that we can't see but we know are there, all right, that if the patient's responding to the systemic therapy, then hopefully that patient maybe has those systemic subclinical sites being sterilized. And the evidence that I know of is that prospectively, those are the only 2 studies that I know of that have ever been done prospectively. Like Dr. DeMoore mentioned before, retrospectively, it's always good to easy to find patients who do well. All right, that's not the fair comparison. The question is, can you prospectively identify a group of patients, treat them aggressively with this? To my knowledge, those are the only 2 studies that have been done prospectively. I would, however, say that for brain metastases, there have been prospective studies, randomized studies, showing that if you have a few number of brain metastases, I think the number was 3 in most of the studies, that there is an improvement in outcome when you compare things like whole brain radiation plus or minus radiosurgery to the lesions. And the same for open surgery. There have been randomized studies of whole brain radiation plus or minus taking out the brain metastases. And in those settings, again, brain-only disease, up to 3 lesions, I think it was, there is an improvement in outcome. Those are prospective randomized studies, which are very encouraging that if we apply that same principle elsewhere in the body, we might have better outcomes in some subset of patients.
And would you say probably most centers can do this type of treatment?
Correct. Almost.
for
This
patients?
is pretty. Most. You want to go to a place that does a lot of this, but most major centers have the capability to do this now. It's easier to treat things that don't move, like a brain lesion, than it is to treat things that move, like a lung or a liver lesion. So it gets harder when you have things that are moving, for obvious reasons.
So I'm going to ask Nancy this question because your lab work. What is your opinion of the current vaccination trials? Maybe Claire might want to comment in oncology.
You mean in terms of the HER2 vaccine?
HER2 vaccine, other things that you think may be promising.
I think that there are some, the HER2 vaccine studies do show some promise. They're not ready yet to be FDA approved. I know that George Peoples, I believe, has a HER2 vaccine that was I just recently read a paper that it is moving forward and showing promise. So really the HER2 vaccination is the only thing that I'm aware of that looks promising at this point. Again, it's not ready to be FDA approved, so it's still in the clinical trial. I'm not aware of any other vaccine trials. Are you?
Well,
I mean,
we've completed a couple HER2 peptide pulsed dendritic cell vaccines.
It's hard to hear, Claire. You might want
sorry.
to
So John Cerroti at UNC and our group did 2 trials of HER2 peptide pulsed dendritic cell vaccines, and they're very small trials, and they look largely at the immune endpoints. What does it do to your T cell response? They're too small to have an ability to look prospectively, did this help more than just standard of care? The group at the University of Washington is doing a lot of HER2 peptide pulsed vaccine work. There's a big new emphasis on immunotherapy. Many academic centers are going to be developing immunotherapy centers. There'll be research centers. So I would agree that there's nothing that I know of that's ready for prime time.
For melanoma, there's been groundbreaking results with immunotherapy with drugs called PD-1 inhibitors that are improving survival in patients with melanoma that have been FDA approved. And so the immunotherapy, because it's working in other tumor types, there's a lot of enthusiasm to try them in breast cancer. Breast cancer might not be the best tumor type because it tends to be less immune prone than the other types of cancers. But there are trials in breast cancer, and those are stemmed from the exciting results that are happening in melanoma.
So here is a question about one of our participants had genomic sequencing done on metastatic lung tumors, but after doing great on a PI3 kinase trial, but what apparently was checked didn't have the PI3 kinase mutation, which always is complicated because maybe we destroyed all those cells. Do you agree with clinical trials which exempt patients that don't have specific mutations or tumor types? And this patient apparently is doing extremely well, this person.
So it's a great question, and it's one of the reasons that I gave that caveat at the end, that nobody has yet shown that because you have this target, you necessarily will do better on this new drug than on something else. And conversely, what if you don't have the target, but you still do well on that targeted therapy? And I think that speaks to the complexity of what we don't understand about the intersection between the pathways. And the second part of your question, I think, is also really good. And as Hai mentioned, with federal funding declining, a lot of the targeted therapy development is being sponsored by pharmaceutical companies, and so there's clearly both scientific but also business decisions as well that go into their development plan. And we've done a number of PI3 kinase inhibitor trials at UNC. Some of those sponsors have restricted the enrollment to people who had activating mutations in PI3 kinase or loss of something called PTEN, so all the known problems in those related pathways that might make you think that that person would respond to this drug. And other companies have left it wide open. And I'm not sure there's much science behind that. It may just be a strategic or philosophical decision. I prefer, particularly in early drug development trials, to have it be as open as possible, because we may learn something from somebody like you, whoever you are, and I'm glad you're doing great.
So here's a great question, and I think all the panel. Why do you think a breast cancer phenotype changes? In other words, why does a patient at initial diagnosis have a hormone receptor positive breast cancer, and then at the time it spreads have a biopsy and it's hormone receptor
Let's all point it
negative
at the same
or
time.
HER2
1,
negative?
2, 3.
You do it. So that's
Next question.
next question. Yeah, that's right. So that's a great. I mean, that's what we're all trying to learn. So, when we do a biopsy and we look at that initial breast cancer, we are looking just at a small piece of it. And let's say you're a person and you had an estrogen receptor positive, HER2 negative breast cancer, okay, a few years before maybe your cancer spread. The pathologist, we look at under the microscope, we look at a tiny portion of the tumor, and we get numbers back like estrogen receptor 62%, which means that the doctor looked at that cancer, and we had a special little chemical stain, and 62% of 100 cells he looked at it, 62 of 100, show the estrogen receptor. So that's a positive test, and we've learned that over the years, a lot of our treatments like tamoxifen, aromatase inhibitors, fulvestrant, are very effective in treating those tumors and shrinking those tumors. But if it's 62%, what about the other 38%? So what we've learned is if you look at a breast cancer, there's a vast array, and it's even a hormone receptor positive, HER2 negative breast cancer. We've learned that it's not like every single cell in that cancer is the same. And so we frequently find that when that cancer spreads, what's probably mutated have been the hormone receptor negative cells in some patients. And that can happen 20 to 30% of the time. So what happens is when we do the biopsy, we find that those cancer cells that are in your liver or lung or bone are hormone receptor negative. And this is one reason why we feel in most instances it does pay to get a biopsy to double check, because we may pick the wrong treatment for the patient if we don't know that. And also sometimes you find a better answer. Sometimes a HER2 negative tumor could become HER2 positive, not that frequently. And it's probably the same reason. When we looked at that little core of cancer, we didn't get a total picture of that cancer. So a marble-sized cancer has 1 billion cells in it. 1 billion. And we put that little core needle in it and get a pencil-shaped area, or we look at it under the microscope. The pathologist can't look at every slice. So I suspect we all believe that cancers have a lot of diversity in their genes, and although we may do well treating the dominant gene in many patients, a lot of the cancer cells aren't responding to that therapy. They're the ones that spread, and they're different. Is that clear? And usually the news is a little bad, like what was good went to bad, like you've lost those hormone receptors, that estrogen receptor, but sometimes it goes the other way. And then the value of biopsies, pertaining to another question, is it allows us to get some fresher tissue and look for some of these new genetic mutations. Sometimes it's hard to track back and find that paraffin-embedded block if it was done 7 years ago. So it allows us getting a better glimpse of the tumor, and perhaps it's more accurate. So I think for many of our patients, one, to establish a diagnosis, and two, to see if there are any major changes that affect our therapy, it's very reasonable and prudent to get the biopsy. And in this age of all the special things that can be done looking for actionable genes, by that I mean abnormalities in genes where we have drugs available that might be helpful, it pays to do it. And maybe I wanted to say earlier, I didn't get the opportunity, that we look at breast cancer as like not a single disease. We know there are different types of breast cancer, but what we're learning now is there are people with stomach cancer that are HER2 positive, and they respond to Herceptin. And probably what's going to happen in 5 to 10 years is we will always characterize tumors as where they begin, but it's really going to be what the genetic component of that tumor is. Because it may be there are some patients with mutations in their breast cancer that are much more likely seen in colon cancer or in lung cancer, like these ALK mutations. And they may be just a few % of patients, but those patients may have an opportunity to really do terrifically well on drugs that aren't standardly approved by the FDA for breast cancer. So we may be looking now at looking at your genes and picking a cocktail for that tumor, and it doesn't matter where it began. It's what it shows now, and I think that's probably what we're going to be seeing. Claire, you want to comment on that?
I agree.
That was long-winded. Okay. There's a question here, in our medical research, is there any knowledge on stem cell research? And I'm not sure from the question if we mean the stem cells that a lot of us or that many scientists feel are related to getting that breast cancer going, that they're the fundamental drivers of the breast cancer. And maybe, Claire, you want to talk about that. Is there anything
I think there's
targeting just stem cells?
There's a lot of research going on. It's largely basic and preclinical from what I've seen on study sections, and you probably have the same experience. I think there are, there are some targeted drugs, if you will, that are called differentiation agents, trying to take something that might be a stem cell and force it to grow to some normal path, or force it to activate a programmed cell death pathway. Nancy, you want to
Yeah, and
jump in?
I think it still is really early. I think on study section, there's always a debate on what is truly a stem cell, what are the true markers to prove it's a stem cell. So I think that there's more research that needs to be done.
Do you know what's, so
Study
study
section
section,
is,
so study section is the group of people who are physicians and scientists and pharmacologists and others, advocates, who sit in a room for 2 or 3 days and review the top 20 grant proposals that a particular branch of the government or the DoD or some
Foundation,
funding agency is going to fund. And so a lot of us, and probably a lot of you, sit on these study sections. So that, anyway, that.
But that always comes up as a, how are you defining a stem cell, and what are your markers to prove that it is? So it's important research, and there's a thought that stem cells may be less sensitive to chemotherapy. So it may be that when you're giving chemotherapy, the non-stem cells respond, but the few stem cells that remain are the ones that are responsible for resistance. And so a lot of people are looking into that. But that's really, it's still early research. And so that's why there's a thought that if you can develop targets for stem cells, if you could combine it with chemotherapy, then you could get the initial cells that are chemotherapy resistant. Okay, but it's still very early.
I've gotten the word from the boss here. Last question. Why do we have to have a large-scale trial to apply genomics to the clinic? Why not use a treatment that addresses an aberrant gene, treat for 6 to 8 weeks, and see if the patient is benefiting? Which I think is something we need this person to write clinical trials for us. We
see this same question come up in big adjuvant therapy trials, which take so many women and so long to complete. And so using preoperative chemotherapy and using the biologic outcome at the end of the 12 weeks of therapy as a surrogate, and there's actually approval of at least one drug on that basis. The issue then becomes, is that, do we believe enough in that early endpoint? If you get 6 weeks of a particular drug and your Ki-67 level drops, does that mean that that drug is going to improve survival in metastatic breast cancer? You need to be sure that your surrogate endpoint is real, and I think that's the big debate. But I think we are, as a community, really working on having trials that are more nimble. But again, you need to rely on the robustness of that interim endpoint, or else you make a big mistake because you put a lot of effort into a drug that may actually not move the ball down the field.
I mean, it's a great question, and there are some studies in women who are getting preoperative chemotherapy. So these are women newly diagnosed, where you have access to biopsy, and if these women consent to trials, there are trials now that are studying new drugs, and they're doing just that. They're studying small groups of patients, adding like a special new agent to standard therapy, looking to see if it's better and if there's a winner. And if there's a winner in a small group of patients, looking in a larger group of patients. I believe that's how it's an I-SPY, I-S-P-Y if you want to Google it. But there are people looking at it. It's a great question. It's a good way to end. And thank you all for your interest. Great questions. And enjoy the rest of the meeting.